All right. Hi, everyone. I'm Jonathan Lee, the Harvard University CFAR Clinical Core Director. Thank you all for joining today. This Harvard CFAR seminar. Seminar is organized by the Clinical Core, which includes myself, Doug Krakauer, the BI Deaconess, Boris Yulg at the MGH Reagan, and Sandy Burchett at Boston Children's. We have two fantastic seminars for you. In part one today, we'll be focused on the design and enrollment of clinical studies. Part two will be held on January 7th, and we'll focus on the secondary use of collected samples. I'm going to put a link to the Harvard University CFAR Clinical Core website in the chat in just a second. But now I'm going to hand it off to Doug Krakauer, who will moderate the session today.
Thanks, John. Welcome, everyone. I'm Doug Krakauer. I'm an infectious disease doctor at Beth Israel Deaconess Medical Center, Harvard Medical School, and Fenway Health. It's wonderful to have you here today. We hope this is useful for all the people who are attending, particularly early stage investigators and laboratory investigators who might want to know more about the clinical trials process. So we're going to try and break that down in a way that's digestible and useful for people who want to go into this field. And I'm going to start by introducing our three speakers today, and then we're going to have the directors of the CFAR and the three speakers as panelists at the end for questions and answers, which you can put in the chat, or you can certainly raise your hand and speak as well. And so we'll invite you to do that, and hopefully this is as interactive as possible as part of the discussion to make it useful for you. So without further ado, let me introduce the three amazing speakers today we're so happy to have today. The first will be Dr. Boris Yulg, who will be talking about the ABCs of clinical trials. Dr. Yulg is an infectious disease doctor at MGH, as John mentioned, who directs the ID Clinical Research Unit and serves as the Associate Director of the Clinical Core, along with me and John and Sandy Burchett. He has a lab at the Reagan Institute, and he works on immunotherapeutic interventions, including therapeutic vaccination and monoclonal antibodies, with an emphasis on translating all these discoveries from bench to bedside. Our second speaker will be Dr. Katie Stevenson, who will be talking about from idea to reality, key elements of launching a clinical study. And Dr. Stevenson is a physician scientist at the Center for Virology and Vaccine Research and the Division of Infectious Disease at Beth Israel Deaconess Medical Center here in Boston. And she has expertise in many different areas, including conducting clinical trials with novel immunogenic interventions for HIV and other emerging infectious diseases such as SARS-CoV-2 and Zika virus. She's currently the site PI and protocol chair for a number of first-in-human studies of candidate HIV vaccines as part of the HVTN, the HIV Vaccine Trials Network, and is also working on large-scale observational studies of respiratory virus epidemiology as her newest area. And finally, we're really fortunate to have Martha Jones, who is going to talk on key considerations for the IRB approval process for clinical research. And Martha Jones is the Vice President of Human Research Affairs at Mass General Brigham and a member of the NIH National Human Genome Research Institute Analysis, Visualization, and Informatics Lab Space Data Access Working Group. And she serves as a member of the Council on the Accreditation of the Association for the Accreditation of Human Research Protection Programs and co-leads the NCCN IRB Directors Group. So she really is at the center of many really important initiatives and programs around IRB and research, and we're really fortunate to have her as well. So without further ado, I'll invite Dr. Yulg to share his slides and begin the talk. Dr. Yulg.
Thanks, Doug. Guess you guys can see that. Okay, great. So I'm gonna talk very briefly about the ABC of clinical trials. Obviously one could talk much longer about all that. This could be quite a complex topic. So I just break it down and make a little bit like a 30,000-foot overview. So in terms of clinical research, really, maybe one of the differentiation is that there's observational studies where you really follow a group of individuals during them receiving their routine medical care. You're interested in a specific phenotype, be it a disease, be it a treatment that they receive. You group them maybe in patient characteristics or the treatments they have, and you assess health outcomes. maybe collect some samples or specimens to study certain biological processes. But overall, you don't really affect the cohort, so you don't assign any particular intervention to these individuals. And so like an example, if we stay within the HIV theme here for the CFAR, is, for example, the cohorts that the Reagan has for many years, studying people living with HIV that maintain viral suppression in the absence of antiretroviral therapy, the so-called elite controllers, and just see actually how they do over many years and study their immune system. And so these are observational studies. What we are focusing more today is on clinical trials. And so these are normally studies where there's an intervention assigned to the participants of the study. They're often randomized, so by luck chosen to be in one or the other group or several groups, depending on the trial design. And there it's really the goal to evaluate medical, surgical, or behavioral interventions and to see actually what the outcomes are from those. Often these studies have a placebo control group, so like a fake treatment group, and new interventions or the new use of existing interventions are being studied in these trials. And an example for that is, for example, to administer infusions on investigational products, let's say anti-HIV antibodies or something like that, to people living with HIV and see how these affect the HIV reservoir. And sometimes these are linked. For example, you do the study here, an observational study, you identify certain antibodies that elite controllers mount, and then you use these antibodies and administer them to people that actually are not elite controllers and see if you have a good therapeutic efficacy by those. So sometimes this goes from one to the other. So clinical trials, just to focus on that, we were looking for two main outcomes here. One is effectiveness of the intervention that you study, and then obviously also safety of the new drug or new intervention that you propose. And then there's additional factors, and that is not supposed to be comprehensive here, but you can define a dose and how much of the product you want to give and how often you have to give it, the drug formulation, being injected, what's the half-life of the drug, and so on. Combinations with other medications, existent or investigational, and then overall look if these therapies actually have an effect on the quality of life. So there's several phases of clinical trials for medical products overall, and many have heard about that. Just briefly, a phase 1 study is a small study with a couple of tens of people that are being enrolled, and this is often the first in-human study of a product. And the primary endpoint is safety. So you want to make sure that your drug that you're testing is safe in healthy individuals. And overall, if you think about drug development, 70% of drugs move to the next phase. The phase 2 now looks at a few hundred people. And there again, one is interested in safety now in a larger group. And often these are dose finding studies. So now you narrow down maybe, between 2 or 3 different dose levels or dose regimens. And efficacy sometimes can be assessed, but normally these studies are not fully powered to determine efficacy of your product. And overall, like maybe a third of drugs move to the next phase after that. So there's quite a down selection process, as you can see. The phase 3 now is really the study where you test it in a larger cohort of participants. And this is really where you look for efficacy. Again, you also assess safety now in more individuals even. And then maybe like a third of that, the drugs then really move forward to ask for registration with the FDA. And then there's a post-marketing study, which is called phase 4. Now the product is on the market, it's being prescribed, it's being given to patients, and you now start accumulating safety data, long-term safety data, and still efficacy data on really large numbers, because some side effects might be extremely rare, and they only show up once you're in phase 4, and the product has been given to thousands and thousands of people. So overall, this is like quite a process. As you can see, you start with many different compounds in the discovery phase. You go through preclinical, and I'm going to come back to that. Then you go through your clinical trial steps, and then at the end, very few of those really make it to approval overall. And if you think about what fails, it's really, and why we have such a low approval rate overall, efficacy and safety are pretty much the big reasons why a drug is not being moved forward. So how do you get from your idea that you have in the lab to your licensed product? And I'm just going to use vaccines as an example here. So you determined your medical need, you know your epidemiology, you know your target population, you know there's the disease burden that you want to protect against with a vaccine. You made your basic science experiments that you have an idea what could be induced by the vaccine to be protective against the infection. And then you go into the preclinical development. So you test this product now in animals for safety and efficacy, and then start making a small batch of product that is produced in a way that it can be given to humans. And so the preclinical assessment is really immunogenicity in animals. So you administer your vaccine to your animal of choice, mice, non-human primates, for example, and you really look that the vaccine induces a strong immune response while your placebo doesn't. But then you can do the same again. You inject your animals with your vaccine, and now you challenge them with your infection that you want to protect against, virus, bacterial. And then you look if your vaccine actually really leads to a reduction of the number of infected animals compared to your placebo. With your placebo, all the animals get infected in an ideal scenario. With your vaccine, none of the animals gets infected. So you have that, and it looks very good. And now you want to really see how much of this vaccine can I give to animals before it becomes really toxic. So you really overexpose your animals in these toxicology studies with significant higher amounts that you would ever prescribe actually for human studies. And this is what gives you a range of what the toxicity of your product is. And then you produce your small good manufacturing practice badge that is then approved to move into a human testing. So you have that. Now you propose to the FDA a new investigational new drug application, and that is being reviewed by the FDA with a protocol, how you want to test that. And once you have that in hand, you can go and start your phase 1 clinical trial. So now you go through your clinical development through the different phases. So just a little bit of lingo, obviously randomized, we mentioned already. Each study participant is randomly assigned to receive either study treatment or a comparator, placebo or active control. So either use the fake treatment, the placebo that looks like your drug, but it's not. Blinded means that either the participant is blinded, so they don't know what they receive, placebo or active substance, or double blind. Again, the participant doesn't know what they receive. The researchers in the clinical trial unit don't know what the participant receives. Only the pharmacist is unblinded. They know actually if the participant receives active product or placebo. And then the endpoints of the study are defined before the study starts to determine what you're looking for. Safety, efficacy, what are your thresholds? And then the study is conducted, and at the end, you determine if you meet those endpoints or not. So the phase 1, as I mentioned, is really to look in a vaccine setting for immunogenicity, but overall it's a safety study and a little bit of a dose ranging study in a small subset of individuals, often placebo-controlled, double-blinded, I just mentioned, and randomized. So you take healthy volunteers. By chance, they're being allocated to 2 groups. Either they receive the vaccine at escalating doses, for example, or placebo. And then after that, you just look at what's the immunogenicity. So what is the immune response the vaccine has induced in people compared to the placebo? And you can see like maybe the different doses induce different strengths of immune responses, but also what's the safety. And there you can see that, and this is all just an example. For example, vaccine dose 3 induced a good immune responses, but barely better than vaccine dose 2. But the side effects were significantly higher in vaccine dose 3 compared to vaccine dose 2. And then you have to define what are you willing to tolerate for the benefit of more immunity. And that actually was done also as an example for the COVID vaccines. This is Pfizer, where they really administered in their phase 1 study different doses. You can see here 10 and 30 and 100 microgram vaccine doses, and overall 30 induced a good immune response, not better than the 100 micrograms. But if you look now at severity of side effects, you can see that actually the 100 microgram here had a lot more severe side effects than, for example, the 30 or 10 microgram. And therefore, then you get good immunity with like the 30 and less side effects than you choose the 30 microgram. And then you go into your phase 2 study. So now this goal is again to look for immunogenicity, safety, and to range your dose. Maybe you do more data to look between 10 and 30, let's say, like for the Pfizer vaccine. And possible you have the ability to look for a little bit of efficacy already, but the studies are not designed and powered to do that. So again, you randomize people into the two groups. And then if you're lucky and it's in a setting where there's enough exposure to an infectious pathogen, you might be able to see a little bit of efficacy data. But again, normally these studies are not fully powered. So safety is really here still often a main factor. So you have that, and think about that, if you think about influenza incidence in the US, which is roughly 8% per season, then to have only 40 cases, so people that actually develop influenza in your placebo group, you would have to enroll 500 people. So these studies immediately become relatively large. So that means you would have to enroll 1,000 people total, 500 in placebo and 500 in active, and that's already like a pretty large study. So if you think about a phase 3 study now, that is really like to look for efficacy. So this is for vaccines, often many thousands of people. For the COVID vaccines, it was between 20 and 30,000 people that got enrolled. Again, people are being randomized, either get the vaccine or placebo. Normally it's a single dose at that point. And then you wait to see how many infections you get in your placebo group and how many infections you get in your vaccine group. And so for the COVID vaccine, as you remember, this became pretty apparent relatively early that there was a split between infection rates going high in the placebo group and being relatively low in the vaccinee group. And so that gave this really high efficacy of the mRNA vaccines in the beginning of like 90%. But that's not always the case. And for some studies, in particular HIV, we often see the curves being pretty identical between placebo and vaccine. So pretty much showing that the vaccine didn't do much and was not protective at all. So you have your phase 3 study completed. You look at correlates of protection. This is specific now for vaccine studies. You might look at different mechanisms for different other products or other outcomes, just to get a little bit more sense what biomarkers you can also use maybe for the future to select your products and to maybe make it a little bit faster in your clinical development and down select your products more efficiently. There was one HIV vaccine study, RV144, that had like a modest but significant effect in terms of protection, as you can see here. And then the sub-analysis really showed that certain immune markers were enriched kind of in people that either were protected or were more at risk of getting infected, had to do with certain IgG antibodies and IgA antibodies and certain Ig-mediated functionalities. I don't want to go into detail, but these are the correlates that you can then look for after you have like these large data sets. So now you have all the data, your safety, your efficacy in large cohorts, and then you can go now to the FDA, and for like a vaccine, you get a biologic license application or a new drug application for other products with the FDA, and file that, and they have half a year to review that pretty much. And then you get, if you're lucky, approval, and the product becomes licensed. Has to go through a process if it's being recommended as well overall, but that's when the product is on the market. So overall, just a very quick overview of the different steps of different phases. It seems quite complicated overall how to go through all that. Normally a process like that takes many, many years. And many products actually don't make it all the way through, but that's just kind of as an introduction for clinical trial testing. And I stop here.
Thank you very much. That was really clear and informative, and I think you did hit the ABCs and plus. And now we'll turn to Katie Stevenson. Dr. Stevenson.
Hi, hello everybody. Let me share my slides. And okay, do they look good? Okay, perfect. So thank you, Boris. Boris and I worked together on many studies, so a lot of that was actually lived experience. So that large overview, I think, gives you a sense of the spectrum of different studies that you may be involved in. And I'm going to just assume for this particular audience that, you're an investigator, you're somewhere in the maybe training phase or a junior faculty, and you're trying to figure how do you fit in here. So for this discussion, I'm going to dive into some real logistics and operations tips that I've learned running a lot of studies, and taking it from the perspective, really from as if you're an investigator and you're jumping into getting a site up and running where you are. So I'm just going to give an overview of kind of from the logistics side, you doing a study. So, first you develop your research idea, and often that's with a mentor, for example, and you sort of pitch your idea, maybe you put an application together, submit it for, say, a K or another type of grant mechanism, and let's say you get funding for that. So we're going to put that all in the bucket of developing your research idea. Then you have to complete the study design. And so we're actually going to spend some time in this area, and this has to do with really firming up all of the details of how this study is going to launch. Then you prepare for your study implementation. So everything is prepared. Everything is sort of finalized for the design, but now you actually have to get yourself ready to go. Then you initiate the conduct of the study. So now you're conducting the study. You complete your study, you manage and analyze your data, you report your study results like in a manuscript, and then you close out your study. So these are sort of the life of an individual study that you might be doing, clinical research study. And I can say my first tip about this is that it feels like when you're running a study that all of the work is in like the first 3 steps, maybe the first, and then once you get to initiating the study, it's like, it's going, it's on. And you just kind of sit back and watch it happen, which is an overstatement, but it just feels sometimes like all the work is in the beginning. So today what I'm going to focus on is steps 2 and 3, which is really about how you can launch your idea into reality. So let's say you've already come up with your research aims, like your specific aims, your hypotheses, your overall study schema, and you've secured some funding to do your study. So the next things that you have to do is make that concrete. And I'm going to spend the most time on this particular part. So you're going to develop a project timeline and milestones. You're going to identify regulatory requirements for your study. You're going to create a budget and what I call mapping your contract requirements. You're going to identify your study team members, your potential research participants, and you're going to make a community engagement plan. So I'm going to just dive into each of those a little bit. So these are all seem simple, but they are very hard. So develop a timeline and milestones. So I recommend that you make a spreadsheet, or even I used to use a Word document template, and I use it with any study that I'm doing. So I kind of keep the same milestones there. You put into that the deadlines for each of the different elements and kind of count backwards from where you should start working on it. Most of the fixed deadlines in launching a study are regulatory. So for example, an IRB that has to meet, and they don't meet every day, they meet infrequently. So you need to know what their schedule is and also the due dates when they want you to submit so that you can be reviewed at a particular meeting. Having said that, the longest activity in my experience is usually contracts. So nowhere in a K application will you ever have to talk about contracts. However, contracts and agreements are actually necessary to perform your work, involve lots of parties, and can be very slow. So this is just a snapshot of a study from years ago of one of my earlier templates for milestone templates that helps you map out kind of all of the pieces. So really, it starts with starting all the way from the top, which is site qualification, which means how you're picked to do a study. And especially for a clinical trial, that's a more formal process, down to when you plan on having your first participant screened and consented to be in your study. So you put all these different elements, and really what locks you in are the meetings, those dates, and then when you need to start it and initiate it. So I think this is just an example of how you can get organized. And often it will unfortunately tell you that you should have already started something by the time you make this plan. So for example, here is like the meeting date for the IRB in this case was September 26th. I expected the approval, hopefully after that, a week or two after that. But to actually prepare for that and submit it, oh, excuse me, that's just for the revision, right? So you also want to plan in like your first review and your second review because you're gonna have two reviews. So that's just to point out that there's multiple steps sometimes with the same project. So you want to identify your regulatory requirements. And Boris went into this in a lot of detail about, what is the type of study that you're doing? So if it's an interventional study, like a trial, that's very involved. That's probably the most involved kind of study that you can do, especially if it's a phase 1 study. And I can tell you, you're not going to be doing that study by yourself. So if you're doing that kind of study, you have a large team that you're involved in. That could involve sponsors, companies, it could involve the NIH. So you will have a lot of assistance with this, but there's a whole pathway of FDA review that's required for that. If it's an observational study, depending on what you're doing, you may need full board IRB review. Sometimes you don't because it's exempt. And hopefully we'll get into that with the IRB discussion. A couple other things to consider for regulatory requirements. So biohazard. So we're doing HIV work, we're doing interventional work, infectious diseases. We often deal with either biohazardous material or a product that could be considered biohazardous. And so those are going to require additional biosafety reviews. And then vulnerable populations, they actually have some additional reviews or extra things that you may need for the IRB. So for example, if you're doing research on incarcerated folks, then you may need an additional person present at the IRB review. Okay, so then you look at the full landscape, and it's actually international, federal, state, and local requirements that you're going to review. I threw international in there because I was just at a meeting that John was at where we were talking about H5N1 studies. And it came up that if anything was found, we'd actually be mandated to report it at a global level. So, oh my God, I haven't thought about the international level. So you have to think of every level of requirements, and then definitely just ask for help from your colleagues or really with the IRB itself, who often provide a lot of assistance for new investigators about these different steps. So the regulatory elements of these different levels, I want to point out a few common things. So like I said, there are international like reporting requirements, but if you have an international site that is involved, and I know a lot of us do work, say in South Africa or other regions, Nigeria for our HIV projects, you do have to think about the local requirements, and those can be pretty extensive and time consuming. For the federal stuff, for FDA, those are really the clinical trials, and you'll need to figure out if your study requires an investigator new drug application or IND. If it does, you are not alone. You're going to be a part of a big project. So I won't go into more detail with that. However, working with the NIH, there are some additional things that are required with NIH funding. So for example, if you are doing an interventional study with recombinant or synthetic nucleic acids research, this sometimes goes to an additional level of review within the NIH. And then one group that I really want you to get to know is the Division of AIDS, or DAIDS. They often are the funding mechanism for a lot of HIV-related studies. Like if you get a NIH grant to do HIV work, you're going to fall under DAIDS. And they have a lot of different training requirements depending on what your study is. And those can be extremely extensive, often required for all the investigators, and just something you're going to want to get familiar with right away. At the state level, that's where you actually see most of the mandatory reporting guidelines. So HIV testing, if you're doing that for your study, you have to look at what the requirements are for you to report those results if you get them. And then you get to local, and that's really where you're going to have the heart of all of your requirements. The Institutional Review Board, of course. Institutional Biosafety Committee, the IBC, that is often required, like I said, for depending on the product that you might be administering. Scientific Review Committee. So if you are not funded by the NIH, like you just came up with this idea on your own and you got funding from discretionary funds, you often have to go through a scientific review within your institution, which is required for as part of the IRB review. And then you have a whole wealth of local requirements. So for example, your clinical research center or the ID research group, the lab has to approve it, the nursing staff has to approve it. So those are all little parts of your milestones that you should put there, and your local institution can help you. Okay, this is the most painful part of research: budget and contracts. And I want to give ourselves time for our other speakers, so you guys can always cut me off, but you're going to want to make a spreadsheet for your study budget. And you're going to include your study staff effort, the investigators, your research coordinators, what supplies you're going to have to purchase for your study, which could be the study kits, but could also be software that you might require. The services, like if you have a phlebotomy service that you need to engage, often we send out lab tests, like even a CBC, you're going to have to pay for that CBC. And then fees, a lot of IRBs and others have fees that they charge. So you need to make a spreadsheet and it has to add up within your budget, within your funding. And then you have to determine what agreements might be necessary for your project. So if you're doing a clinical trial, that will be a clinical trial agreement. Again, you'll have other people helping you with that because involves a lot of legal parties. Oftentimes, you might need a research cooperative agreement. So even for an observational study, if you have multiple partners, you'll need something like that. You might have to set up a vendor. So those are more simple agreements, but still have to be done. And then if you have collaborators, you might have to set up a subcontract. So those parties that are involved in that run from like your tech ventures office to your office's sponsored programs to your legal department. And you're going to want to figure it all out because, like I say here, is the secret killer of all your timelines, or all these agreements. Okay, so you want to identify your study team members, and that's just a picture of some of my study team to show how friendly everyone can be. But for your clinical staff, depending on your study, you may need extra levels of expertise. So maybe you have to, like maybe it requires a doctor for some of the clinical trials. Other studies, maybe not, maybe a research nurse or nurse practitioner, and then your clinical research coordinators. And then often if you have the funding, you may be able to have a community engagement staff that's dedicated to that activity. And then for your study, you want to identify your potential participants. So you're going to really consider your target population for your research aims. So that's number one, right? What's your research question and what's the population that's going to help you answer that? And within that, determine if you need to recruit any special populations. And there are a lot of different types of special populations, but three big ones that come out often are children, pregnant persons, and then also prisoners or incarcerated individuals. They have special protections. They may have to go through additional reviews. They often require different types of consent forms. And so that's, again, something you'll need to determine. And then I put here to make a conscious effort to match local demographics at a minimum. And that's really because I don't have nearly enough time, and you would need 2 hours to talk about community engagement work for increasing the diversity of your patient participant population. As we all know, Black participants, particularly Black individuals with HIV, are often underrepresented in the studies that we're doing. Women with HIV are often underrepresented. So you have to make this conscious effort. And that is a much longer discussion, but you should put it into your planning at this stage. And you make your recruitment plan. So figure out where you're going to find these folks. If it's at venues like tabling at universities or health fairs, or if you're going to have to do more directed peer referrals, like from physicians. For a lot of our studies involving people with HIV, we often have to work very closely with clinics to get those direct referrals. So you'll make your community engagement plan. And I say this because you really can never start this process too early. You probably want to be doing it in between studies to some extent, because you want to identify your community partners and the stakeholders. So those are HIV support groups. Like there's a group we work with a lot called Healing Our Community Collaborative. It's a HIV and women support group. Or AIDS Action, Boston Pride, Fenway groups. These are all key partners, and then look at prior studies and see like where did they go wrong and what populations were not represented and how can you fix that. And sometimes you're going to need a community advisory board. A lot of HIV studies that are funded by the NIH often require them. So if you don't have the resources to set up your own, you can partner with others that may give you a chance to talk about your study with them. So then after all of that, you are able to prepare for your actual study implementation. This is really like almost go time. So it really helps to prepare what I would call a study operations manual, but it doesn't have to be as formal as that. We do that for clinical trials, but it could just be for you a set of your standard operating procedures that you're going to use for your study. Because you may have investigators and other people in the study, and you want to show them like, this is how we do it. So for example, phlebotomy, we're going to draw some blood from people. Well, which tubes should we do? How much? What are our transport conditions, etc. So you want to make a little collection of SOPs or a manual for that. You're also going to create your data collection forms and your source documents. For big trials, you'll have help with that. For your own studies, that's a lot of work. You want to do in-services with ancillary groups that are going to help perform your study. You have to order your supplies, figure out where your study is actually going to happen. That's often getting permission to use your clinical research center. And then you have to train your study personnel. Some of it will be required certifications like CITI and other things. Some of it will be study-specific trainings. Okay, and that will bring you to that's sort of where we're at that point. When you've done everything, you're really ready to start. And then I put here the steps that come after is that now you start it, you initiate your conduct of the study, you get your data, you report your manuscripts, and then you close out your study, which are all topics for further discussions. So I will end there so we can move on to the IRB discussion. Thank you.
Thank you, Dr. Stevenson. Really great practical tips and also super clear. So for our final talk, Martha Jones, are you ready to go?
I am ready to go. Let me get my display here for you. And thanks so much, Katie. That was a great introduction to hopefully some of the points I'm going to bring up. So thank you. So I am going to, like my colleagues here, talk very quickly at a high level about IRB and preparing for your IRB submission. Let me start with really thinking about what is the purpose of the IRB. I know many people start off with a thought that the IRB is a barrier to research, but really we have tried to change that and think about it as a collaborative effort with our researchers, because we really do want to enable the ethical conduct of research and support you in the great work that you're doing. Our key goal, of course, is to approve research, making it compliant with all the federal regulatory and institutional requirements. But that's really not only from the standpoint of protecting our research participants, but protecting you as researchers to ensure that the great work you do is not out of whack with the federal requirements about research. So Katie referred to some different types of research review. This is what it looks like from the IRB perspective. The IRB can review in several different ways. And starting with the least onerous, they look at projects and decide whether or not you're actually conducting human subjects research. If it's determined you're not, then you don't have to deal with the IRB and those regulatory requirements. There might be other requirements you have to deal with. Once it's determined that what you're doing is human subjects research, then there's kind of three pathways. One is called exempt, and this term gets a little confused with some other terminology in the regulations. But in this context of human subjects, it means that your research fits into a category of research that's defined in the federal regulations, and that you really have minimal regulatory requirements if you fall into one of these categories. The kind of workhorses of IRB review, however, are in the last two categories here, which are going down a path of expedited review, which first and foremost, to qualify for this kind of review, the study has to be determined by the IRB to be what's called minimal risk. In that situation, if it's determined to be minimal risk and it fits into a category, again, defined in the federal regulations, then it can be reviewed by a single IRB member. That's what expedited means here. It isn't really meant to mean fast, although we hope it is a fast review. That single person can make a decision to approve the study. All regulations still apply to those studies, just like the studies that are more than minimal risk, but the review process is through that single individual. That individual cannot disapprove a study, however, they can only approve studies. And then really the category that applies to clinical trials that we're focused on here today is full board review. Most of the clinical research, especially if there's some kind of intervention, as we've been discussing, is going to be considered to be more than minimal risk and have to go through a full committee review process. So I want to give you a little background as far as what the IRB is looking for, because it will really, if you understand this, it will help contour all those steps that Katie just went through, from the deciding on an idea for research through preparation and getting ready for conduct of your study. To think about from the very beginning, what are the ethical considerations and regulatory considerations that I have to weave into my protocol development and the conduct of the study. So we start with what's called the Belmont Report, and you may have heard this before, which defines the ethical background for what the IRB does. From the Belmont Report, in the regulations, there are a set of what are called criteria for approval. So when the IRB reviews your study through either that expedited or full board path, they have to look at your application and decide whether it meets these specific criteria. It's not just a kind of whim of the reviewer whether those are met or not. They have specific checklists that they are going through your application to assess. And those criteria for approval, you tell the IRB about those criteria by filling out your application. So let's go through this. This is the Belmont Report. The three fundamental principles are respect for persons, beneficence, and justice. And those become operational through ideas like getting informed consent from participants, as Katie mentioned, protection of vulnerable populations such as children. The IRB has to look at your protocol and assess the risks and potential benefits of that from the participant's perspective, and understand the risks of your protocol. And the justice principle helps us think about who should be included or excluded from a research study. What recruitment methods are you using? How are you going to disseminate the information from the study? And as Katie talked about, community engagement would fall into this justice principle as well. So with those as the background, as I mentioned, we move into how do we apply those to the criteria for approval. So these are those criteria that the IRB, either the single IRB reviewer or the full board, must consider, and how they relate to those principles. Again, they have to make sure that risks are minimized and that they're reasonable in relation to any anticipated benefits of the research, that you're selecting subjects in an equitable manner. For research that is more than minimal risk reviewed by the full board, you have to get informed consent. What is included in that informed consent is again dictated by the regulations and institutional policies. They will require that the consent document be signed if it is a more than minimal risk study. The IRB is also looking at how are you monitoring the data and the activities to ensure safety of participants. What are the provisions for protecting the privacy of participants and confidentiality of their information? And then making sure again that those that are considered vulnerable are protected in the conduct of the research. So finally, as you're thinking about preparing your protocol and your application for the IRB, all of those pieces of the criteria for approval and the underlying ethical principles are are worked into our IRB applications and the questions that we ask there. The main one that you all would be dealing with from the standpoint of a clinical trial would be what we call our intervention interaction protocol. Different IRBs might have different applications. We use an electronic tool called Insight, which is a place where you can submit your IRB application electronically, and the IRB uses that to review and communicate with you as far as your IRB review process. So with that as background, I'm going to highlight just a few things to get you started on your IRB application and what to think about. Importantly, you need to understand the regulations that are going to apply to your research study. The IRB has basically two sets of regulations that they have to enforce and review by. One is called the U.S. Health and Human Services regulations. Those are managed by the Office for Human Research Protection. Sometimes you'll hear people refer to OHARP or OHRP. These regulations apply if your activity involves humans and if it's funded by a federal entity. So those regulations technically do not apply to research if there's no federal funding. But we, like most IRB, apply these basic regulations to all of our research, regardless of whether it is federally funded or not. The U.S. Food and Drug Administration, or FDA, also has very similar regulations, although some of them are different just because of the type of research that they oversee. Their oversight is not based on funding, so even an unfunded study could come under the regulatory oversight of the FDA. They kind of take on oversight of a study if it involves biologics, drugs, or devices and human subjects. In lots of cases, both sets of regulations might apply to your research. And certainly if you're doing clinical interventional research, those FDA regulations are likely to apply. So before submitting to the IRB, it's really important that you start with the basics of completing any human subjects education that's required by your entity. Here at Mass General Brigham, we have some internal education that we have developed, which involves a course that we call our boot camp course, which gives you some basics of conducting research. And we also have a good clinical practice specific course to take as your basic required education. But we also have a menu of several other items or courses that you can take as demonstrated here on our research hub. I've given you a link to that. And you can look at courses based on pathways designed for human subjects research by an investigator educational pathway, animal research, et cetera. So I would encourage you to take a look at those. We also have an IRB 101 course that you can take. It's important that whoever your IRB is, that you read and understand the IRB's policies and guidances. I've put a link to ours here, but those are going to contour how you perhaps design your study even. Examples would be there's guidance or policy here around who needs to obtain consent, whether it's the investigator or a study coordinator can do that. There's rules and regulations around texting, on recruitment procedures, and there's also guidance, for example, on actually how to fill out your application and submit attachments in our tool called Insight. We also have a lot of services to help you, as Katie kind of mentioned. Reach out to your IRB. We have office hours every other week where you can just drop in and talk virtually to our staff. You can ask for a specific consultation, and we'll work with you at any time. We have an email service also for submission questions. Very importantly, walking through the steps that Katie did, it's important that you do not submit to the IRB until your study is fully designed. All your documents are ready to go. Everything is ready and in place. and you would be ready to actually start recruitment as soon as you get your IRB approval. If you're not at that state, you're probably submitting too early to the IRB. If you're still developing materials, it's likely to be materials that the IRB actually needs to review and approve. And by submitting to the IRB before that point, it's actually only going to slow down the review process, not only for you, but for all of your colleagues, because the IRB needs to spend more time going back and forth with you to get the information they need to assess those criteria for approval. So start out by writing a very detailed protocol. We have a template for that, again, that I've linked here. Explain what you're going to do in a logical, orderly manner and provide appropriate detail to the IRB. Don't just write what you think the IRB wants to hear, because the IRB is going to hold you to what you write and make sure that you conduct your study based on what you've written in your protocol. Again, make sure all your materials and processes for data collection are complete. Anything to be seen or read by the participants needs to be finalized. The IRB can only approve final versions of documents. Really conduct a feasibility review, and I think Katie walked you through this really nicely. Make sure you have any resources you needed, that any agreements are in place, and that you have permissions for conducting the research wherever you plan to do that. And certainly identify any other required or ancillary reviews or approvals that you might need. Again, maybe it's the pharmacy department or other nursing, et cetera, that you might need to get approval or interface with first. So just wanted to provide you with, again, some information about our contact and how you can get help from our IRB if you need it in your preparation. Thanks.
Thank you so much. That was a wonderful talk about such an important part of this process. And we're going to transition to the panel discussion. And the first thing I wanted to do was to ask Dr. Raj Gandhi and Dr. Ingrid Bassett, who are the co-directors of the CFAR, for just some brief words about other places people can go to find more resources beyond the clinical core and what we've talked about today.
I'll just start by thanking all of the people who spoke and say that there's more than can be covered in such a brief time. So really what we're here to do is to offer resources. Dr. Bassett had put up a QR code around some of the resources through the Harvard CFAR. But I think it could be easily encapsulated by saying in clinical training, we're told never to worry alone. And in research training and clinical trials, you have a team of people here to help you. I think reaching out to some of the people on this call, if you have a specific question, to other people, just to find out where the resources are. We won't be able to really cover all the resources, but what we can commit to doing is linking you to people who can help. The CFAR has a number of cores. We have good connections with the clinical trials networks around the country. Many of those clinical trials networks offer samples and data that you can build on, but really the main point is reach out to us and we can help guide you and really connect you. So I'll stop there, and maybe Ingrid, if you want to say anything, and then we'll take the questions.
Yeah, sure. I think just to say the Center for AIDS Research has a lot of tools for all these different phases of preparing your idea and performing the research. We have trainings, we have consultations, we have funding for pilot studies. And really our focus is on early stage investigators. And so please do feel free to reach out to us. And I encourage you to think broadly about what HIV research is. It may be that you do research about tuberculosis or hepatitis C or substance use disorder and infections. You know, we have funded research in all of those different areas. So please think broadly about that. And this QR code can help you get signed up for our listserv. So I'm happy to take questions and be on the panel.
Great, thank you again. And thank you to all the speakers. Just going to the chat here, there were two comments and one comment and a question to mention. Ronald Bosch says, in addition to contracts, there may be other delays around things like data use agreements, especially interinstitutional ones, or around international shipments. That's a great point. I think Katie showed her cards about headaches with potential contracts and delays, but there are many other aspects that can slow things down. So that's a really helpful comment. And then Allison asked a really insightful question, an important one about how you might navigate the tension that could exist between developing a novel scientific idea that might compete with or create some friction between an early stage investigator and their mentor or mentors. And curious, Raj and Ingrid, as the directors of the CFAR, what would you comment about, what kind of advice would you say about how to navigate these situations should they arise?
Yeah, I mean, I can say that, you know, obviously, if you're still in that mentoring relationship, having that frank conversation with your mentor around your ideas and often, and I would hope in most instances, your mentor is going to want to promote your career and work out an arrangement where you do your trials. They may be a senior author, or they may step aside depending on how distinct it is from what they're doing. But yes, this is obviously a challenge, and the bigger the stage is, the more challenging it can be. You know, Ingrid, if you want to reflect on your advice in this arena as well. Yeah.
Yeah, I mean, I think I agree completely with your point about communication with the mentor, I think is really key. I think having multiple mentors so that you're working at the intersection of a few different people's work can also help to create a separate niche or pathway for you so that you aren't necessarily just doing what your mentor's doing, but you're doing something with like a slightly different twist. And that's where I think co-mentorship can be very helpful for having you bring something new to maybe to your primary mentor's table that isn't exactly the same as what they're doing. But I think the communication's important. And you know that in fact, the mentee's success is the mentor's success. And hopefully you have a mentor who can see it that way and not feel threatened. But I do think that is a risk.
Thank you. Those are great responses. Anyone else in attendance who wants to ask a question of the panelists in the last minute or so, please feel free to put it in the chat. If not, John, you had one question before we close for the three speakers.
Yeah, I mean, I think that we have a really illustrious panel here who have all run clinical studies and clinical trials. I'm wondering, what did you all find to be some of the most useful resources when you were learning to run clinical trials? Was it specific courses or classes? Was it your mentor? Was there some other resources that our attendees should know about?
I can say
I
very
just want to say
briefly.
one quick thing,
Yeah,
which is
please.
I feel the biostatistician is very, very important. I meet with the biostatistician early and often in the life cycle of a clinical trial or a clinical study. And I really, really find them very helpful for clear thinking. And I really recommend them as a resource. I think I learned a lot from them in terms of how to do a study.
I'll
I was
just add,
gonna
there's so many
say,
different dimensions
oh,
to clinical trials. This will be brief. That having, and the mentor doesn't always, isn't going to always be at your own institution. And this clinical trials group, what I learned the most is by having a co-chair, someone who guided me through the process. It really revealed kind of all the different ways to solve problems. And so I think that's probably the most valuable experience that I had is in this instance, having a mentor. I was at another institution who served as an experienced co-chair. But sorry, Katie, you should get the last word.
it's similar in that I learn best by doing. I've taken a lot of courses in my life, but I think the what I have found the most helpful is when a mentor or a colleague has allowed me to be on a protocol team. And be able to sit in from the very beginning, all of these steps, and be there for all the problems all the way through, and then move up another level, maybe to a co-chair position. That's how I've learned everything.
And the people on this call can make those opportunities available, I think, whether again, it's data, lab samples that we'll talk about in the next iteration in January, or introducing you to teams that you can be part of. I think that's our role.
So with that, I think we'll close at the hour and then advertise, as Raj just mentioned, that there'll be part 2 of the series right after the New Year's. And so we really encourage people to attend, bring bring your friends, bring your colleagues, and this is really meant to serve your needs. Bring your questions as well. Thanks to everyone, the speakers in particular, all the panelists and the attendees, and we look forward to seeing you after the new year. Have a good, happy holiday.